Welcome to my channel. Today, we're going to explore one of the most remarkable scientific breakthroughs of our time, how artificial intelligence cracked the protein folding problem. This is a story about microscopic molecular machines that are essential to every form of life on Earth. These incredible structures have evolved over billions of years to perform an astonishing variety of vital functions. They're called proteins, and for over half a century, scientists struggled to understand how they work. But in a stunning development, researchers used AI to solve a key piece of this biological puzzle, and in doing so, won the 2024 Nobel Prize in Chemistry. Proteins first appeared on Earth at least 3.7 billion years ago. Since then, nature has produced countless variations of these molecular marvels, each designed for a specific purpose. They come in all shapes and sizes, and they're constantly at work inside every cell of your body. You can think of proteins as the workhorses of biology, the molecules that actually get things done. So how did AI crack this 50-year-old mystery? That's exactly what we're going to uncover. This is the inside story of how a team at DeepMind used artificial intelligence to solve the protein folding problem, a breakthrough that opened the door to a new era of biology and earned 3 pioneering researchers the highest honor in science. So what exactly is a protein? At its core, a protein is a chain of smaller molecules called amino acids, strung together like beads on a necklace. There are 20 different types of amino acids, and each one has unique chemical properties. The specific sequence of amino acids in a protein determines everything about how it will function. When a protein is first assembled inside a cell, it starts out as a long, unfolded strand. But here's where the magic happens. This linear chain doesn't stay flat for long. It folds up into a precise 3-dimensional shape, like a piece of molecular origami. And it does this every single time with remarkable accuracy. That folded shape is what allows the protein to do its job. Proteins perform an incredible range of functions in your body. Enzymes are proteins that catalyze biochemical reactions, including the digestion of your food. Antibodies are proteins that protect you from pathogens and disease. Structural proteins give tissue its shape and strength. Muscle proteins power every movement you make. Signaling proteins help your cells communicate with each other, and transport proteins move molecules throughout your body. They're truly the molecular machinery of life, and understanding how they work has been one of biology's greatest challenges. In 1969, a biologist named Cyrus Levinthal made a puzzling observation that became known as the Levinthal Paradox. He calculated that even a small protein with just 100 or so amino acids could theoretically fold into an astronomical number of possible configurations. We're talking about 10 to the power of 300 possible shapes. That's a number so large it defies comprehension. If a protein had to randomly try each configuration to find the right one, it would take longer than the age of the universe. Yet here's the paradox. Real proteins fold into their correct shapes in less than a second, sometimes in just milliseconds. How is this possible? How does nature find the right answer so quickly in such an impossibly vast search space? This contrast highlights just how remarkable biological folding really is. A brute force computational approach would be hopeless, searching through trillions upon trillions of possibilities. But nature has somehow evolved proteins that zip right to the correct answer almost instantaneously. Understanding this process became one of the central mysteries of modern biology, and solving it would require entirely new ways of thinking about the problem. The mystery behind protein folding became known as the protein folding problem, and it consists of 3 closely related questions. First, how does the sequence of amino acids encode the final 3-dimensional shape of a protein? Second, what are the series of stages along which a protein folds? And third, how can we computationally predict the 3-dimensional structure from the sequence alone? This third question is where things get really exciting for us in the AI and machine learning world. If we could predict protein structures computationally, we wouldn't need to spend years and hundreds of thousands of dollars determining each one experimentally. The protein folding problem became the domain of a field called structural biology. Structural biologists study protein shapes, formerly known as structures, to figure out how they work. If we know the three-dimensional structure, we can begin to understand how these molecules behave and function. The potential applications are enormous. A better understanding of protein structure could lead to cures for diseases linked to misfolded proteins, like Alzheimer's, Parkinson's, and sickle cell anemia. It could revolutionize drug design, allowing us to create medicines that precisely target specific proteins. It could make biological medicines cheaper and faster to develop. The stakes couldn't be higher.
For decades, the primary way to determine protein structures was a technique called X-ray crystallography. In 1957, biochemist John Kendrew used this method to reveal the first atomic protein structure in history, a groundbreaking achievement. The process works like this. First, you purify the protein and concentrate it into a crystal form. This is often the hardest part and can take months or even years. Then, you fire a high-intensity X-ray beam at the crystal and rotate it. The X-rays scatter off the electrons in the protein, creating a diffraction pattern on a detector. That diffraction pattern is then transformed by computer into a three-dimensional electron density map. From here, it's like solving a jigsaw puzzle. Researchers fit amino acids from the known protein sequence into the map to produce a structural model. In the early days, scientists actually built physical models using balls and sticks. Later, computational tools accelerated this process. But here's the catch. The effort required is enormous. Determining a single protein structure can cost around $100,000 and take a couple of years of a PhD student's time. It's an enormous investment for just one structure. By the 1970s, researchers created the Protein Data Bank to catalog all known structures. Today, it contains over 200,000 entries, but that's still only a tiny fraction of all proteins in nature. Newer techniques like cryo-electron microscopy have helped, but the fundamental challenge remained. These experimental methods are slow and expensive.
In the 1960s, biochemist Christian Anfinsen made a discovery that would later earn him the Nobel Prize and fundamentally change how we think about protein folding. In a series of elegant experiments, he took a folded, functional protein and denatured it, which means he caused it to unfold and lose its function. Then, when he reversed the conditions, something remarkable happened. The protein refolded itself back into exactly the same shape, restoring its function completely. This was revolutionary. Anfinsen had demonstrated that all the information needed for a protein to fold into its three-dimensional structure is encoded solely in its amino acid sequence. You don't need any other biological machinery to guide the folding process. The sequence alone contains the complete blueprint. And here's why this matters for computation. If the sequence alone determines the structure and no additional cellular machinery is required, then theoretically we could use mathematical and computational methods to predict the structure directly from the sequence. This insight opened the door to an entirely new approach, one that would ultimately lead to AI solving the problem. But getting there would take another 50 years of painstaking work.
Let's look at how proteins actually fold. The process happens in stages, each building on the last. It all starts with the primary structure, which is simply the linear sequence of amino acids, like letters in a sentence. But this chain doesn't stay linear for long. First, local regions of the chain start to form secondary structures. There are two main types. Alpha helices are coiled, spring-like sections where the backbone twists around itself. Beta sheets are formed when strands of the chain line up side by side and link together, creating pleated sheet-like structures. Both of these are held in place by hydrogen bonds, which are weak but numerous chemical connections. Next comes the tertiary structure, where the entire chain folds into a complex three-dimensional shape. This is driven largely by interactions between the amino acid side chains. These side chains have different chemical properties. Some are attracted to water, others repel it, and their interactions with each other and their environment guide the folding process and stabilize the final shape. Some proteins go even further, combining with other protein chains to form quaternary structures, larger complexes made of multiple subunits working together. Throughout this entire process, the protein is essentially searching for its lowest energy state. It's following the path of least resistance, settling into the most stable configuration possible. You can visualize this as a ball rolling down a funnel, eventually coming to rest at the bottom. That bottom is the protein's native functional shape. By the 1990s, scientists recognized they needed a rigorous way to measure progress on the protein folding problem. Enter CASP, the Critical Assessment of Structure Prediction, a biannual competition founded by John Molt that would become the Olympics of protein prediction. The idea is elegantly simple. Experimentalists share the amino acid sequences of proteins they've just solved but haven't published yet. Computational teams then try to predict the three-dimensional structures from those sequences alone. At the end, the predictions are compared to the real structures and scored on a scale of 0 to 100. The criterion for success has always been to reach a score of 90. At the first CASP in 1994, the computational predictions were, frankly, terrible. One experimentalist famously rolled about laughing at how wrong the predictions were. Nobody could predict protein structure accurately. The researchers would joke about how they still had job security for another 10 years. But CASP served an important purpose. It organized the community around a single metric and allowed everyone to see what worked and what didn't. One researcher particularly inspired by that first CASP was David Baker. He saw an open problem worth dedicating his career to. His lab developed a program called Rosetta, which tried to simulate the actual physics of protein folding computationally. It was a physics-based approach, trying to model all the fine-grained atomic interactions that happen during folding. For more than a decade, researchers made incremental progress using physics-based methods, but no one achieved a major breakthrough. Then, in 2016, something remarkable happened in a completely different domain. DeepMind, an AI company owned by Google, demonstrated that their AI system, AlphaGo, could defeat a human world champion at the ancient game of Go. This was considered impossible just a few years earlier because Go has more possible positions than atoms in the universe. During that famous match, DeepMind's founder, Demis Hassabis, had a thought. He remembered playing Fold-It, an online game created by David Baker's lab where anyone could try their hand at folding proteins. Hassabis wondered, could deep learning, the technology behind AlphaGo, also solve the protein folding problem? In 2017, theoretical chemist John Jumper joined the DeepMind team to find out. They developed AlphaFold, using machine learning techniques similar to those used for image recognition, but wrapped in protein-specific knowledge. They trained it on the 136,000 structures in the protein databank at the time. In 2018, DeepMind entered AlphaFold in CASP 13 and won. Their predictions were better than anyone else's. But as Jumper put it, they were the best team in the world at a problem the world was not very good at. The scores were improving, but still not where they needed to be. So the team went back to the drawing board, determined to completely redesign their approach for CASP 14. AlphaFold 2 represented a complete redesign of the system, built with protein knowledge integrated directly into the neural network architecture. Here's how it works. First, you input an amino acid sequence. The algorithm then searches genetic databases to find similar protein sequences from other organisms. These related sequences are aligned in an array called a multiple sequence alignment, or MSA. This captures evolutionary information about what nature has tried and what has worked. Next, AlphaFold generates a pairwise representation, essentially a two-dimensional matrix encoding the spatial relationships between every pair of amino acids in the sequence. You can think of this as a map of which parts of the folded protein will end up near each other. These two representations are then fed into the evoformer, a powerful transformer-based neural network. The evoformer uses a technique called self-attention to efficiently extract meaningful patterns from the data. What's remarkable is that it sets up a kind of conversation between the evolutionary data and the geometric beliefs about the protein. The information flows back and forth, continuously refining both representations. Finally, the refined information passes into the structure module, which calculates the actual three-dimensional geometry and produces coordinates for every atom in the protein. The prediction is then cycled through the whole algorithm multiple times, refining itself with each pass. AlphaFold also outputs a confidence score for each part of the structure, telling you how certain it is about different regions of its prediction.
CASP-14 arrived in 2020 during the pandemic, with everyone working remotely and on lockdown. The DeepMind team submitted their predictions and waited anxiously for results. When the scores started rolling in, they could hardly believe what they were seeing. Many of AlphaFold 2's predictions achieved scores above 90. The target that had seemed unreachable for decades was suddenly being exceeded regularly. John Molt, the founder of CASP, sent an email that would go down in scientific history. He wrote that DeepMind had performed amazingly well, both relative to other groups and in absolute model accuracy. It was really a shock. Researchers kept asking, can it really be that good? What's going on here? Several factors contributed to this breakthrough. DeepMind built their understanding of protein physics and evolution directly into the neural network, what's called inductive bias in machine learning. This allowed the system to learn extraordinarily rapidly from the data. And the protein data bank provided a data set that was uniquely well-suited for deep learning, with high-quality, well-structured examples that follow consistent physical rules. By July 2022, DeepMind had released structure predictions for 218 million proteins, nearly every known protein in the world. They made the code freely available, turning it into a resource for the entire scientific community. The press proclaimed the protein folding problem essentially solved. Every experimentalist now tries AlphaFold as a first step, sometimes saving years of work.
In October 2024, the Nobel Committee made it official. David Baker, Demis Hassabis, and John Jumper shared the Nobel Prize in Chemistry for their groundbreaking work on protein structure prediction and design. It was recognition of a 50-year journey that had finally reached its destination. But the story doesn't end there. In 2024, both DeepMind and the Baker Lab released next-generation tools. AlphaFold 3 adds diffusion-based methods to predict not just protein structures, but how proteins interact with DNA, RNA, and small molecules. The Baker Lab's ROSE-TT A-fold all-atom can predict the structures of complex assemblies containing both proteins and other molecular components. These advances are opening doors to applications that were previously impossible, designing drugs that precisely target disease proteins, creating enzymes to break down plastic pollution, engineering proteins that capture solar energy more efficiently, developing new sustainable materials. The possibilities seem endless. As the scientists themselves put it, there's so much more to be understood. This isn't the end, it's the beginning. We're entering a new era where AI and biology work hand in hand, where computational prediction accelerates experimental discovery, and where the molecular machines of life are finally giving up their secrets. If you found this video helpful, please like and subscribe to the channel for more content on AI and scientific breakthroughs. Thanks for watching.